thoughts there?
Well, I'm not sure. I'm sure Anthony has more to say about it than I do, but I mean, I think there is this continued march away from animal data, and also I think, as Pascal was saying just a few moments ago in our pre-discussion, also potentially this shift toward including more real-world evidence, um, as opposed to a confirmatory trial, which I think are both interesting chips away at the pharma model, you know?
Yeah, absolutely agree. Anthony, have you had any thoughts about this? I mean, this is relatively new, but it's probably not new. It's been coming for a while.
Yeah, I mean, I think what I would say, I'll speak from both sides of the table here. We've all been hearing about phasing out animal testing forever, since
Yeah.
animal testing came in, right? Since Charles
Yeah.
River started a mouse house 60
Yeah.
years ago, it's been on the way out. And I think, to misquote somebody I can't even remember, that the rumors of its demise are exaggerated. But I think the FDA gets this too, and probably the most important thing to take home from their statements is that they express it as a roadmap, right? Which
Yeah.
implies there's a way to go in the future. And I think the time frame they mentioned was 3 to 5 years. Now we all know that timelines get doubled. So I think in 6 to 10 years, yeah, I think I actually would like Lee or somebody else to expand on his comment about the increasing attention on real world evidence, because I think that bears not only on preclinical data, but also on clinical data.
Yeah, yeah, absolutely. And I think we'll definitely be touching on some real world evidence, to a little bit extent in this session, but also in future sessions as we go. So, hello everybody. It's wonderful to have you join us here on Advanced Therapies Think Tank. It's amazing to see everybody here, and it's wonderful to be participating again. So, if you are new to Advanced Therapies Think Tank, we are a solution-based platform really set up for experts across the advanced therapies ecosystem to enable us to tackle these really big challenges head on. The format is we skip the primers and we dive straight into the real questions, because we know you're working in the space, you don't need all the preamble, you've got a really good idea and you just want to get to it, and we don't want to waste people's time. Every episode is really built around a single pressing topic, and we bring in expert voices from the industry, from payers, regulators, and patient-facing roles, with the aim to really challenge the assumptions and create real actionable ideas. Our faculty, as always, helps us to shape these discussions throughout the season, and we'll close every session with key takeaways, and Lee is with us today to help out with that. Now, before we dive into the really good stuff, none of this would be possible without our season and our session sponsors. So I do want to take a minute to really give a huge thanks to our season sponsors, BCA Advanced Therapies Network, as well as to our sponsor for this session, Vitalant, for supporting this work. So one of America's largest non-profit blood and biotherapies healthcare organizations, Vitalant sees every day how critical it is to accelerate the delivery of advanced therapies to patients who need them most. Too often, outdated, innovative models show progress and raise barriers, slow progress, excuse me, and raise barriers to access. So for this reason, Vitalant is keen to help facilitate the discussion today between big pharma, cell and gene therapy industry, regulators, and others. This is very much a not-for-profit. No one who runs this program in Advanced Therapies Think Tank gets a salary. We do this for fun and for the ecosystem. So we don't have any sides. We're not trying to protect or promote anyone in this. Now, on to the main event of today, and I'm really, really excited to introduce the faculty and speakers. So today our faculty members are Angela Volstead, Anthony Davies, and Farzan Fazaneh. And we're very happy to also be joined by our invited guest, Pascal Touchon. In this session of Advanced Therapies Think Tank, we're really going to get back to basics, but not in a way you've seen before. We're going to inspect the foundations of how we develop advanced therapies and discuss what really needs to change and how it needs to change in order to really fulfill our commitment to patients, of developing truly life-changing therapies in record times. As many of you are very aware, the current biotech innovation model is based on the pharma model for small and also for large molecules. It so often doesn't take into account the unique characteristics of advanced therapies as living medicines. And so by requiring the use of these outdated development models, there's a real risk that advanced therapies are at a developmental disadvantage. This results in time-consuming, expensive, and potentially not any better therapies for really desperate patients. Now, as we get started, please don't forget there's a chat function. Use it, discuss with each other, raise reactions. There's also Q&A capability within the Zoom mechanism platform. So please put in questions. They can also be upvoted. So if you really like a question, you can upvote it, and we will have a session at the end to address these questions. The faculty, when they're not speaking, will also engage with some of the chat. So feel free to really get involved. Okay, so exciting times now. We talk about, as I've said, the biotech innovation model, but really, what does this mean? Is it really an innovative model or just an adaptation of how pharma has developed things for years and years and years? How can we move away from this and develop true innovation that shows the incredible advantages of advanced therapies, but whilst ensuring the safety of patients that we're aiming to cure? So let's start off with you, Angela. Biotech is supposed to be exciting, fast, and efficient. But why is it that we still don't have a proper innovation model that we can be proud of? What can we learn from the current model, and how can we improve?
Sven, thanks. It's an excellent question, and one that we're going to have a lot of exciting discussion around today. Let me start by saying this. Biology isn't code. So unlike digital technology, biology does not scale neatly. So when we're talking about innovation models, what we need to bear in mind is what we're trying to do here is for each disease area, each modality, each patient population, we have unique challenges that we face. So coming at this from a mindset of looking for standardized models, that's going to be hard to replicate from other areas. We also face challenges that are structures that we operate in or legacy. Biotech inherits a lot from big pharma, and the biotech industry is obliged to follow rigid clinical development pathways that have been established for decades. Functions across the sector are siloed, and governance in general, both within corporations as well as the regulatory agencies, tends to be risk-averse. That has an influence on how capital is allocated to development, and it certainly influences how smaller and larger players are able to develop their products. VCs, for example, as we all know, push short timelines and are very much focused on getting phase 1, phase 2 readouts. What happens after that tends to then slip a little bit out of focus. And that sort of long-term system innovation that would be required to really talk about an innovation model that addresses the potential of advanced therapies sort of comes to a bit of a halt there in this constellation. So what we see is there are these bursts of innovation that take place in research and early development, maybe scaling into phase 1, 2, but we don't have models that are really durable from end to end, from research to commercialization. And that fragmentation in the ecosystem between the regulators, the biotechs and the pharma companies, academia even, and the payers. So across the ecosystem, we're really facing something that is best described as silos, legacy silos, with pretty weak integration. If you think about the therapeutic potential that we want to be able to translate from discovery through the clinic into market access. Now, biotechs traditionally have been built around the hero molecules, and the question around how to translate fundamentally new ways of manufacturing drugs, especially bespoke ones, is something that deserves more attention to platform technology that can be used across the spectrum of disease indications and therapeutic areas. So there are some players in this space who do push the platform mindset more than the individual biotechs. But I would say if we're going to talk about innovation and trying to identify what would make for a new innovation model, we have to think in a lot more integrated fashion. From academia right through biotech into pharma, are we actually trying to work in the same or very similar ways that can scale through the different segments of the system, rather than having what we have today, which is a lot of breakage and slippage along the way, that academia works in one way, then come the biotechs, try to scale this out a little bit, and then from a big pharma perspective, sort of translating that into a marketable product becomes the challenge. So in a nutshell, I think what we should be looking for a new innovation model is very much more partnership-driven, collaborative approaches across all sectors of the research, academic, and the industry space.
Brilliant. Thank you so much. That's a great start. I really like that partnership and bringing everyone together and kind of trying to get everyone aligned. So kind of thinking about that, Farzan, I'm going to come to you next. Now, you spent a lot of time in academia and now more recently in industry, and you've been thinking a lot based on many of our conversations going late into the night. You've been thinking about how do we do things better? And so what would you say are the areas that we should already be innovating on in the preclinical space? Let's start with that and move from there.
Yeah, in fact, before maybe focusing on innovation, Sven, there is, let me start somewhere that you might not expect me to want to start. There is a lot that has already been innovated that we don't explore and exploit therapeutically enough. So my analogy, forgive me if I have already used this before in some of our late night conversations over a year or two, that the wheel was in fact invented thousands of years ago. But we failed to recognize that they could be fitted to suitcases until after the landing on the moon. And there is a lot of examples of that kind. For instance, safety switches, as in gene therapy or cellular therapies, that would guard against unwanted side effects in case they happen. Now, those need to be ideally auto-regulated so that it would knock out the one cell that has malignantly transformed before it becomes a disease in the clinic or a problem. And to do that, it's conceptually, biologically quite easy because there are a lot of tumor tissue context dependent regulators, promoters, enhancers, post-translational modifications that could be employed, and we don't employ them well enough. And similarly, we don't adequately address issues, again, with autoregulation, which involves, for instance, responsiveness in a context-dependent manner to environmental cues, say, when we want to address an autoimmune disease or graft versus host disease in tissue transplantation. So whilst innovation is, of course, incredibly important, and I'm being facetious, suggesting some of these are just sitting on the table waiting to be deployed, I think thinking in the right context and going back to some of the discussion we had a little earlier, actually, across the whole group, in the context of any disease, we need to be sure that we address the right disease in the right context, even for phase 1. And again, to use an adage that I've maybe overused in the past, the fact is that phase 1 clinical trials in healthy volunteers may not entirely demonstrate either efficacy, let alone the toxicity that may be associated when it is used in the correct context. And I spent a little bit of time talking to regulators, and one of the favorites from the MHRA is at least that mice lie and monkeys don't tell the truth. Hence, emphasizing the point that we need to be in the right context. So,
Brilliant.
yeah.
No, that's really, really fascinating. And absolutely agree. I think we need to be make sure that we're well positioned for this. And I love that last bit about the monkeys and the mice. So true. So let's kind of move on a little bit. And Anthony, so you spend a lot of your time evaluating and also supporting various manufacturing organizations and helping them to really become better. So what are you seeing as you support this pursuit of innovation, and where are the areas that, one, you're seeing improvement, and two, you're not seeing fast enough improvement in innovation?
Yeah, I mean, making these things is hard and complex. I've just actually got stuck into a nice LinkedIn discussion on the subject of well-characterized, biologics, cell therapies, gene therapies, and so forth. And I've got a fairly Marmite, polarizing view on that. I'm going to sort of dodge your question initially and then come back and answer it. Honestly, a lot of contract manufacturing, well, first of all, I can talk about contract manufacturing organizations, and I can talk about in-house manufacturing, or try and smush them together. Sven's not moving, so I'm either his internet's frozen or I'm just gonna pick one.
I mean, I think we have to do both, right? I mean, they're both an
Okay.
integral part of the
Let's start with
ecosystem.
CDMOs. Legit, Sven. Oh, there he goes. He was frozen. So CDMOs, we're at a no news to anyone. We're at a Darwinian point. Okay, a lot of them are dying. It's really tough. We're in that, it's a very cyclic thing, capacity and demand and supply. What was it? 8 years ago, there were articles in the New York Times saying there wasn't enough capacity. Now they're dying off like flies. Okay, so at the same time, there's a sort of flight to quality in that market, and CDMOs, contract manufacturing organizations, are really trying to differentiate themselves. And a lot of them are trying to say, hey, we're the AI CDMO, we're the automation CDMO, this is why we're special. And honestly, my advice to them, which is almost always somewhere between disregarded and hated, is first, just be a really good CDMO. Okay, just focus on manufacturing. AI and automation, all that stuff is sort of going to happen, but you are not going to win this battle by being the most AI CDMO ever if you can't manufacture basic stuff. Again, not super welcome message usually, but I'll stand by it, okay? Because it's hard enough as it is, and AI is not going to save your ass instantly. Neither is automation going to change your cogs overnight. There's massive capex and time and all that stuff there. So I think that's my sort of, stick to basics is still my advice there.
Yeah.
In-house manufacturing, I think that has in some ways the luxury of not really being in this sort of competitive maelstrom. And I'm very sympathetic to CDMOs, by the way, at this moment, that in-house manufacturing is not suffering the same competitive maelstrom events that contractors are, but by the same token, they still need to get it right. They still need to get the basics right, and then and only then, I think, should manufacturing organizations look at the highly innovative things that are out there. I've got more sort of controversial stuff to say on the subject of how to fix manufacturing, but you'll just have to stay watching to hear that later.
Excellent. I like that. I love that. And I think start with the basics, get the basics right, and then you can start getting fancy. But until that stage, don't even try. I love that. Fantastic. So let's move on a little bit. Pascal, as a reformed therapeutic developer, what were your observations of this supposed innovation model, and where did you really seek to break the mold? Perhaps then maybe if I can also ask you to opine a little bit on the news we saw this week of those 3 new guidelines from the FDA one on regenerative medicine expedited programs, which is possibly something you can talk to, given your experience, and also, from Europe, some about post-approval, and then on smaller populations. So, really keen to understand, do you think they're really going to move the needle, or is this just some more yada yada?
Yeah, I think first of all, I'm very pleased to be here. I'm going to talk on my own behalf as a personal individual who's been working on that for many years now, not under the name of the company I've been working for in the past. But clearly, when we started all that idea to bring an advanced therapy to the market, we were confronted at the time at Novartis with three key issues from the innovation model. One was the clinical trial themselves, because we were dealing with some rare, ultra-rare type of population. And no, ultra-rare is not defined, because rare in the US is less than 200,000 patients. When you deal with a few hundred patients, 1,000 patients is what I call ultra-rare, and it is different from 200,000 patients. But you are dealing with small population, and you have to deal with that. And then you are dealing with a modality that is not prone to double-blind type of situation. Even you cannot have a sham easily, and it's very challenging to have a true double-blind there. So that created some changes on the innovation model, because at the time, the innovation model had to be a double-blind randomized controlled trial. So how do you adapt to that? Then the second challenge was around the modality itself from a CMC point of view, because with a living drug like a cell therapy, like Kymriah was the first ever cell therapy approved and cell gene therapy approved, you were dealing with something that was very different. And therefore, you had to redefine CMC and what you call quality, what you call the key CQAs, what you call potency. Everything has to be redefined completely from that idea that you're giving something that is going to be a living drug, expanding, circulating, trafficking everywhere, and doing something in the body. So we had to redefine that with the regulators, and I'll come back to that in a minute. And the third aspect was more on the commercial side, because we are bringing therapy that were potentially curative and certainly one-off therapy. So you had to redefine, reinvent completely the payer's view on how to pay for a drug. So these three key challenges were very significant, and they've been well addressed in general by many people and many products. There have been a number of products approved, not enough to our taste and not fast enough for the patient. But there have been a number of approvals there. So the question is, how can we improve on that? Because clearly there have been a lot of changes in delays, in extra cost, or even in complete response letters coming from the agencies or the equivalent outside of the US that have created some significant issues for investors, for the operational team, and for the leaders of the biotech and pharma industry to address more of the unmet need with this type of therapies. But I'll start saying there's been a lot of progress. Now, you ask me about what can we think about the guidance. I mean, guidance is always useful. They're always a bit late and a bit outdated when they come out. It's interesting in that particular guidance from the FDA on trials, when they lay out, I think, 6 type of studies that could be adapted to this type of cell and advanced therapies, cell and gene therapies, as well as smaller population. And when you look at these 6 trial design that I mentioned, and just for memory, it's about the adaptive clinical trials design, the externally controlled studies, the one single arm trial, Bayesian trial. All of that has been used already by all the products that have been approved. So it's not something really new. What is probably new is the fact that everything is laid off. Maybe the one on the master protocol design is a bit new, but all the rest has already been used and successfully so, and sometimes with some changes. So where this is helping, I guess, is to set the scene for what could be done and what could be discussed with the agency, in that case the FDA, to make sure that there is some room to maneuver that is more adapted to this type of rare small population, but also this type of therapy that, as I say, do not allow to have a proper RCT design for many clinical trials. The other aspect which I find interesting personally was the possibility to rely upon real-world evidence into the post-approval setting to a level where that might completely replace some confirmatory trials. I mean, to be seen whether that's really followed with action. But that's certainly encouraging to see that, because again, going into confirmatory trials, especially for a small biotech company, is always a very, very challenging endeavor, especially when you need to treat a relatively small population. And again, you cannot do an RCT in an easy way. I mean, RCT are difficult. I mean, we tried at one of my company to do a randomized control trial in phase 2 in multiple sclerosis, but that was a real challenge. And again, it's not possible in all the different cases, all different situations. So again, these guidance, these draft guidance are going in the right direction, but it's not enough to really break the innovation system and adapt it to advanced therapies. And we need to have more flexibility, especially on one key aspect we'll discuss, I guess, later, which is around CMC. Because I still think that on the CMC front, despite the number of guidance, despite some progress, despite a number of approvals, we still having to deal with an environment that is more influenced by small molecule and biologics than by what is a cell therapy.
Fascinating, fascinating. Thank you very much, Pascal. And absolutely agree. I think what we saw in the early days of gene therapies that I was involved in also is many of these innovative approaches. There was just a lot more discussion with the regulators about could they, couldn't they, would they, wouldn't they. Whereas now it's being laid out. So that I think is, yeah, it's a teeny tiny small step, but it's definitely a step in the right direction. And so we should be grateful for all of those moves. So we're starting to see some movement, some changes, some separation. What I want to move on to now is a slightly different topic and thinking about vectors. So vectors, as we use them, are somewhat unique to the advanced therapy space, not just viral vectors, but the various non-viral modalities also. So we can kind of bring them all together. And within the therapies we develop, they very often perform a critical role in creating and making these advanced therapies. But they are often, particularly from manufacturing, some of the biggest causes of headaches and delays. So Angela, from your perspective, why do you think, and we discussed this a little bit in a chat we had a couple of weeks ago, where you talked about vectors as being the new drug substance. Give us a little bit of insight as to why that is, and how should this approach drive a new and better way of developing therapies that's more targeted?
Yeah, Sven, I want to build on what Pascal was just saying. The hurdles very much do lie in the CMC space and the understanding of how we do CMC and how we regulate CMC, and what are the limits with what we're trying to do in applying legacy ways of regulating CMC. Traditionally in biologics, and small molecules, the drug substance is the active ingredient. It's the molecule itself that is what drives the drug or the therapy. Now, for gene and cell therapies, the medicine, of course, is delivered and enabled by a vector. And viral or non-viral, as you say, it is what makes the therapy arrive at its destination. It is the functional payload carrier, whether that's an AAV or a lentiviral vector or any non-viral vector technology that's used, but the manufacturing of these vectors is key to any of the advanced therapy modalities that we're talking about, and they determine if the therapeutic transgene makes it to its destination, where it needs to go, where it is to have the therapeutic effect. So, of course, the vector itself is never going to be the drug substance, the active ingredient. But without the vectors, any of them, the genetic information and the changes that we're trying to effect cannot reach their destination. So by themselves, they cannot become the drug substance on their own. So it's a little bit of a help to think about the vectors being so essential to advanced therapies that we may want to think about them the way in legacy modalities we think about drug substance. Without the drug substance, there is never going to be a drug product. And so that's a little bit the tension that I see here, because the vectors have to be consistent, and they are like an anchor to any of the advanced therapies. Their quality and the reproducibility and the yield with which they can be manufactured define the potency, the safety, the scalability of the advanced therapy manufacturing processes, just like they do for APIs in a more traditional biologics or small molecule space. So that's very key to anchor that. Now, a lot of the time we talk about the therapies themselves, what we aim to do through the transgenes that we're aiming to deliver. However, without the vectors, none of this can be accomplished. And that makes them, for many different reasons, if you will, the key bottleneck, the key ingredient in any advanced therapy, and the manufacturing capacity that is often spoken about. And we heard Anthony speaking about this just earlier, is something that's gained a lot of attraction in the space, a lot of attention. Is there too much? Is there not enough? Who is the right partner? But I want to fully agree with what was said earlier, is stick to the basics and get this right, because without that, vector being correctly manufactured, consistently manufactured, cost-effectively manufactured, the advanced therapy that depends on it really runs into trouble. And that's clearly something that's happening across the space with a lot of players, is that this is a genuine bottleneck. It is also the science. In this case, it's not just manufacturing, it's the science. Understanding how vectors are manufactured, how vectors can be consistently quality controlled is a bit of an art, and it is something that it doesn't necessarily get overlooked. But I think in order to drive advanced therapies forward, we need to have a lot more focus on vectors, because they are the key differentiator for this particular modality space. And it is a space, it's not one size fits all, but there have to be broad discussions about improvement in vector design, which is the right type of vector for the type of therapy I'm trying to develop. Do I want a virus? Do I want a non-viral? What is my payload size? What immunogenicity profile have I got to take into account? What kind of disease area and type of disease development am I trying to intervene in? And that dictates really what the requirements are for the vector to be chosen. I think sometimes that the vector choice comes after the fact rather than before. And that is one of the shifts I would like to see, is that before we select the therapeutic approach, there is a consolidated evaluation of which is the right vector to use to get to where we're aiming to go. So that's something that I could see being able to drive perhaps more nuanced development approaches, and maybe even assessing vectors against each other before jumping in and saying we've got the therapy in hand already. So a platform first approach, really.
Great. No, I absolutely love that. And I can definitely echo it. So many young companies I see, choose a vector. And when I say why, they're like, because that's the only one we have. So, completely agree. I think this is fascinating. So I think we'll get more into that discussion about those vectors. And I'm also going to ask Pascal to talk about that. But first, I want to go to Farzan. So Farzan, you obviously, have got a teeny tiny little bit of experience with vectors. And so hearing about the importance from Angela, I'm hoping, you've been dancing a little jig about that. But we still struggle, as Angela has said, with cost of goods, with manufacturing at scale. And these are some real challenges. So, what are the solutions that you are seeing and that you are trying to see to address the scale problem? And how will solving these really help us live this innovation dream that we have?
Yeah, that is such a lovely question. And I'm so fortunate to come in immediately after the comments that were made by Anthony, Pascal, and Angela right now. So it's a privilege to talk about this. The only problem is, Sven, you may have to shut me up as appropriate, because I may go rabbiting on forever. So I have two immediate responses. So manufacturing. Generally speaking, I'm speaking as a CSO for a small CDMO, and as also in my other capacity as chair of molecular medicine at King's College London, that as soon as you start talking about manufacturing and amount of virus that is needed, even when there is adequate attention to all other aspects, the first knee-jerk response is that, I don't know, if 10 liters gives me X and I need 2X, I need to have 20 liters. Whereas it doesn't occur often enough that, well, if 10 liters gives me X and I need 20 I need 2x, maybe I should do something to increase the manufacturing capacity, the manufacturing, not just scale, not volume, but that each cell makes more of the same vector so that the output is increased without expanding the volume or the size of the vessel or the incubator and so on. And to that extent, there is a lot of new and a lot of published literature already on how one may use a whole variety of strategies to increase the manufacture of various vectors. And in the company I work at VirusCell, we employ all of those, including small molecules that have been into clinical trials for other applications. We now add them to cells that are producing the virus, and we get, 10 times more virus put out. So that is using a 10-liter manufacturing to get the equivalent of 100-liter output. The other is that Angela spoke very elegantly about choosing, and others mentioned, choosing the right vector for the right application. So, so often we don't use the optimum vector. When we think of lentivirus, for instance, a vector close to my heart, we immediately think of an integrating lentivirus with the risk of insertional mutagenesis and with the payload that it has, ignoring the fact that some 20 years ago, non-integrating lentiviral vectors were described, which have, for many applications, obviously not for all, some of the advantages of AAV without the limitation of the payload being 4 kb, instead being 9, 10, 11 kb. And issues of that kind, there is a lot of literature now on needing to express multiple genes in the context of one vector, be it viral or not viral. And then the issue of capacity and the relative control of these components becomes important. Particularly, imagine if you have, for instance, imagine cytokines that would increase the efficacy of a CAR T cell. And imagine that if the target cell, the CAR T cell that is generated, was expressing those cytokines at constitutive levels. It's under unregulated promoters. Well, there may be a whole variety of consequences because of systemic expression of those cytokines, instead of those cytokines being expressed under the control of regulators that would only allow expression in the context of having bound the right target cell and not the wrong target cell. So what was described by colleagues speaking before me is really important, really relevant, not just for manufacture, but for design, but for the also the choice of vector. People have already described lentiviral vectors that are episomally maintained and therefore may not be just suitable for non-dividing cells, as in maybe, or less dividing cells, as in neuronal or cardiac tissue applications, but also for dividing cells because they're episomally maintained. So this strategy that it would be very convenient to have one size fits all is like my old boss, Pete Borst, many decades ago used to tell me that complex problems have simple but wrong answers.
Fantastic. Absolutely amazing. Farzan, thank you so much. I would love us to keep going and turn this into a 2-day seminar, because I always learn so much from all of you. But that was very enlightening, and I just had this idea of pulling levers, because I think we often think of one lever very often for these things, and it's about how do we adapt all those different things. And so with that kind of thought, Anthony, I'm going to come on to you. So we've heard it from Farzan about other ways of scaling, but what are the other roadblocks when it comes to cost, and what are the other ways we should be thinking and we are thinking about improving those costs, and how can we really utilize that innovation and bring that to the forefront to do things better for patients.
Do you want me to go like full nuclear? Is this the point in the discussion?
We want solutions. So Lee is waiting with his pen poised for solutions, so we can get this out to everyone.
Lee's on mute, so let's just have a party. Look, when I started Dark Horse, I gave this talk called "It's the CMC Stupid," which was riffed off the Bill Clinton election campaign. It's the economy stupid, and then I changed that presentation's title to "It's the cogs stupid." And I think Fars, despite your career for vector, that's still a massive thing. Okay, so there's three. I'll now present a detailed analysis of cogs for vectors. That it's people, places, and materials. Right, the people are expensive because until automation and AI blah blah takes over the world, you need frequent intervention by skilled humans who are expensive. You need a big old facility that's expensive to cook up, and materials. Yeah, plasma's expensive. I know there are cell lines and all that stuff, but raw mats are also really expensive. All right, heads up, everyone. Put your phones down. There's a place in the world where all of this has been solved, and that is mainland China. Locally sourced raw materials are many-fold cheaper. Engineers, I'm not talking about percentage changes here, I'm talking about fold changes, okay? And if anyone's going to bleat on about how there's contaminants in them because China doesn't have its act together, they sorted that out over a decade ago. That's ancient history. The raw mats are fine out there. So the raw mats are, I don't know, X-fold cheaper, 3, 4. The facilities, there are so many federal and local incentives out there. No tax for 5 years, free trade zone, blah, 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 blah. If you do it in the right place in mainland China, which is probably either Suzhou or the Greater Bay Area, and by my impeccable Chinese pronunciation, you can tell I've been spending some time there recently. Then it's free. Okay, that part of it is basically free. The people, I'm not going to over exaggerate here, but salaries are conservatively, well, they're probably 3-fold cheaper than Boston. Let's just put it that way. And we've done some preliminary analysis, which I sort of half-heartedly put on LinkedIn and got a lot of hate mail for, that the cost differential is conservatively 5-fold and potentially as much as 10-fold. Most of the hate mail was from people at CDMOs, but I won't comment on any potential conflicts there. And so for supply chain, which, as you all know, recently Dark Horse is very interested in, you can freeze it and put it in a bottle. If you can freeze it and put it in a bottle and ship it, that's fine. I'm not suggesting you make CAR T cells there, okay? I'm not doing intercontinental shipping for leukopaks. That's a dumb idea. But for anything you can freeze, anything bulk and drug substance you can freeze in a big bottle and pop on a plane or even a boat for that matter, that's fine. There are a few flies in the ointment here. There's geopolitics, there's tariffs, there's a few other odds and ends floating around that need to be tidied up. But I will tell you, I take a long-term view. I think this is a Steve Jobs theft. The long arc of history is going to go straight over the current geopolitical tiffs and tariffs. Anyway, tariffs, 100% tariffs, that's 2-fold. I'm talking about 5-fold. So fine, we'll pay the tariffs. But whether we're ready to entrust manufacturing of those that sort of material there, we manufacture all of these things over there, and so far World War III hasn't broken out. And finally, my final point on this rant, which you'll either move on from rapidly and I'll never be on this program again, or we'll discuss, The final point is, if you think that they're not qualified or they're not smart enough to manufacture complex ATMPs, nope, they are. Been there, checked it, check.
No, that's brilliant, Anthony. Thank you very much. I really appreciate that. And I think we are less so, but we've been very stuck on the China is lesser than the West, and I think that's a very false approach that's been propagated by people and organizations with a hidden agenda, let's say. So I think, really fascinating. And I've just been writing some notes, and I think, maybe the whole China issue and how to work in China is and should be the topic of another advanced therapy think tank in the future.
I'm down. I think it is. And there were solutions there. I would come with solutions, not problems there.
Yeah, that's absolutely brilliant. No, I think definitely something to talk about more in the future. So Pascal, coming on to you, vectors really important, but vectors, even, to Angela's point, they are not the be all and end all. There are other components around this in the space that we're working. So what are the other areas that are really screaming out for innovation, and what are these innovations that we need to be bringing in yesterday?
Yeah. I think outside of vector, I think this, at least for cell therapy, the cell themselves. And I think we still in the early phase. I remember very well the July 12, 2017, when we had the ODAC of Kymriah. And if you spend some minutes to look at the slide from the FDA at the time, they were approving a product based on 63 patient study, single-arm study, with a kind of mixture of cells that nobody understood what it was into it. There was a mixture of T cells, not many B cells, mostly T cells, CD4, CD8, effector, memory, you name it. Some of them transduced, some of them not. I mean, a big mixture. And it is a product. And the other, like Yescarta and others, have not really changed that. Now, what is changing now is not only having new type of cells beyond the T cells, but also to have a better refinement of the type of cells. And at the time, we started a program at Novartis that was called YTB323, which was the idea of not only doing a more rapid manufacturing, but also being able to select central memory cells. And that product is still in development. But the point I'll try to make with that product is that they were able to dose it at 20 times less than Kymriah and having at least the same efficacy and possibly even a better safety. So again, that is answering to improvement in innovation, but also to cost of goods, by the way, because by definition, if you give only 12.5 million cells per patient instead of giving 250 million, that changed a lot for an autologous, and even more so if you move to allogenic, the cost of goods and the ability to deliver an innovation. So the cell is the key, because at the end, that's the one that's going to do the job, of course, with the vector when they are transduced, but they are going to do the job, and the way they're going to expand, traffic, persist is going to make a difference there. Now, this being said, the innovation is, to me, the key answer to the particular aspect that Anthony mentioned, and it's very true that there is something happening in China that is massive. I was lucky enough to be confronted to that many, many years ago. Same year, 2017, I traveled to Shanghai and to Nanjing to meet a very small company that was not known, that everybody thought it was a sham, and that company was called Legend Biotech. And I was the first Western company to visit them and see where they were making Carvykti. And this has become now the big blockbuster of the field, and it's going to grow again. The point I'm making here is that at the end of the day, whatever, when you talk about cost of goods and cost of people and so on, that's one aspect of the story that is very important for our industry, for sure, for cell and gene therapy. But the most important is innovation, because what these guys did was not having, at the time, and still today, not a very new way of manufacturing this BCMA CAR-T, but it was just to have a particular binder that proved to be more efficient, and then a right type of development and investment into that. So the message I would like to convey that the cell is key, and there are ways to innovate on the cells and on the way you manipulate these cells that will make a big difference and can leapfrog clearly all the issue and cost of goods. And if we have time, we can talk about in vivo CAR-T, but that's also another part of the story.
Yeah, fantastic. No, that's really, really interesting. And, that must have been an amazing trip to China to see Legend, in those days. And
when was that? When was that, Pascal? Sorry.
June 2017. right after
That's
ASCO, because there is this guy from Xi'an that presented data at ASCO on this patient with 100% complete response. Nobody believed him. So
yeah.
I went to China to see where it was coming from.
Yeah,
That's
wow.
awesome.
That was a bit of a miss for Novartis, but anyway, let's move on.
Yeah, but I should say my failure was not to license it because J&J was preferred. But anyway, that's another story.
Brilliant. No, that was really, really exciting times. And your point about the in vivo, yeah, absolutely. So for everyone, listening, I think it's on the 6th of December, we have an in vivo versus autologous therapy session where we'll be exploring a lot of those topics. So I am very aware we've been getting into some really good detail here, really great discussions. We've blown our schedule right out of the water. And gone over time dramatically. But I do want to, address one last teeny tiny topic about platforms. And so I'll ask each of you to answer, but try and be relatively succinct so we've got some time for audience questions. So we've talked about, various mechanisms, vectors, the cell, being able to improve it, but there are other areas we should consider. And, there was some mention before about platforms. So let's sort of try and focus on some of those areas. So Farzan, starting with you this time, really help us align with this. What is a platform? Really, what's the definition of it? It's a term that's used very broadly, but I suspect there are a lot of definitions in our space. Can you speak to some of those, and how should we think about it so we're all aligned?
For me, a platform in the context of viral vectors and gene therapy, and for instance, generation of CAR T cells, is a vector system in which similar controls and construction components are used to deliver different therapeutic to deliver on different therapeutic objectives. So this vector that targets CD19 and another that targets CD20 or 22, they could all carry the same background, and therefore, as a platform, save on some of the safety efficacy studies, but not forgetting that the payload is the bit that does the job, and therefore that cannot be part of a platform. One cannot argue that CD19 has been tested and demonstrated to have efficacy, and therefore the safety and efficacy can be extended to CD20. So, but a lot of other components can be used, and similarly, safety switches, auto-regulation can be used as components, like, if you like, as in Legos, in the construction of different therapeutic products.
Wonderful. That's really, really helpful. Thank you very much. And it started making me think about that Lego song, but I'm not going to get sidetracked. But I am going to go back to Pascal. So, regulatory hurdles, lack of harmonization, unclear guidance, or lack of early enough guidance have all been blamed on delays. What have really your experiences been with various regulators, and how can we help them to serve companies and by extension patients better when thinking about platforms?
Yeah, I think that there are two aspects in your question. One is how you can harmonize the various regulators around the world, and to make sure that as a global developer, when you're a biotech or you're a pharma and you develop a product globally, you can make sure you don't duplicate work and you really don't delay access for patients, which is the ultimate goal, is to go fast to patients there. But that's a very challenging one, and there are a lot of think tank and activities try to harmonize that. On the other hand, there's also ways to optimize your development in terms of taking advantage of some differences. I mean, there are some countries where you can go a bit faster on first in human for CMC, like Australia. You have China, where you can go faster to patient in general. And we see more and more companies trying to go first in China, or try to do a Chinese development in parallel to another type of development in another countries to be able to have faster access to patients. You have difference between EMA and the FDA in terms of the CMC aspect and the way they look at that, but of course the clinical trial. So ultimately, one of the challenge we discussed earlier on in this particular field is the need to have patient data. that not only we want to decrease the use of animals in experiment, but the animal data do not mean much for us in these advanced therapies. And we need to go to patient fast, and we need to find ways to use this patient data to validate not only the particular type of modality you're using, whether it's the vector, the cells and everything, but better understand how your product is behaving in patients, and use that even on the CMC front sometimes, because based on ICH guidelines, sometimes when you have variability in your product, if you have patient data that confirm that variability, this type of variability that you have is acceptable from a safety and efficacy point of view, then you can go and present that to a regulatory authority. So the point I'm trying to make that in parallel to harmonization, which is a goal that is very important to avoid cost and duplication, there is also this ability to leverage some differences and get as soon as possible to patients with a product that is safe enough you can use, but get this patient data that will help you to create value for the patients.
Brilliant. No, really, really interesting, and some very good ideas there. I, absolutely love that. So Anthony, coming to you, one of the buzzwords that we've been hearing about recently is analytics. So what are really some of the massive improvements that you've seen in analytics and data that, will honestly accelerate how we innovate, not just fill out your AI scorecard for more AI slop, but what will really help accelerate these products and make them more cost effective? And then, I'll give you this chance to tell us your hopes and dreams for an innovative analytical development, the companies out there to help them to innovate better.
Yeah, I think that is so key to this field. Another LinkedIn scuffle I've got involved in is this well-characterized thing. But I just want to answer in general terms first, Sven, and I want to bore you all with a short lesson in history, because I think there was an inevitability about the analytical characterization thing. In the 1950s, there were two competing theories for what made proteins. One was a colloidal theory, where there was sort of suspension of amino acids, believe it or not. And the upstart theory was the peptide theory, where proteins were comprised of linear chains of amino acids. And during that time, Max Perutz and John Kendrew and a few others just gambled on the polypeptide theory and said, well, if that's true, they should have a rigid structure that will deflect X-rays in a predictable manner, and that was the field of X-ray crystallography and a Nobel Prize. But when he was trying to determine the structure of proteins, it was still controversial whether proteins even had structure. In biologics, I'm old enough to remember when the concept of a well-characterized biological was laughed at. Just decades and decades away. All right, they're proteins, they flex, they have post-translational modifications, they're trimmed, they're all over the place. Just not going to happen. And in less than 10 years, biosimilars was baked into the world's pharmaceutical supply chain. Celltrion, other companies were just founded and built on that thing. So don't tell me we can't have well-characterized cell and gene therapies. It's going to happen. Plan on it, okay? Have your patent lawyers plan on it, because your biosimilars are going to clean your clock at some point. Have your manufacturing long-term planners plan on it. It's going to happen. Now, when's it going to happen? Now I'll get a bit more lame. I don't know. There are some really interesting problems to solve. I'll tell you one thing: deep sequencing and generating terabytes of DNA data, probably not the answer, even with AI. So don't go down that road. One of the really critical things is potency assays. And I'll just highlight a couple of points to give some stuff to chew on. For gene therapies, especially ones, which is most of them, addressing monogenic traits, it's relatively easy. You know what the gene's meant to do. does your therapeutic do it? Okay, it's meant to be a methyltransferase. It's meant to be a whatever it is you're stuffing in the vector. Cell therapies, especially those, and again, shoot me on the sort of frilly edge of cell therapies, like stem cell or mesenchymal, all-knowing, all-singing, all-dancing stem cells. Sometimes they can work. Okay, Mesoblast just proved me wrong on that. Okay, sometimes they can work, but we often don't really know why.
Mm-hmm.
All right, and they're getting to the root of the mechanism of action and getting a proper potency assay. Your potency assay is just intrinsic. It's the soul of the medicine. It's the potency assay. So that's on my wish list, is some new, probably academic science, Farzen, some new academic science determining how to determine how cells do what cells do. Okay, that's what I want for cell therapies.
Brilliant. That's really, really helpful. And
Absolutely.
I think it almost feels like we could do a whole session on potency assays, given the headaches that they have caused in the past. I am very, very, very aware that we've just hit the top of the hour. We haven't even gotten to our single audience question, other than the great chat that Rafael's been having. But I want to give Angela a chance also just to comment on this last one, and thinking, bringing this whole discussion round circle into thinking about collaboration. You talked about collaboration, and no one can do this all on their own right at the beginning. So, we know that the biggest pharma can't do everything on their own, as much as they've tried. So how do we expect smaller biotechs and startups to do it all? Tell us about, what is the innovation that will work to support collaboration, and tail us off for today.
I'll give it a shot anyway, right? I'm not sure I've got the answer. I would never expect a biotech to be able to do it all. If big pharma can't, why should a small biotech be able to do that? I think the essence lies in a mindset that consolidates around the notion of a collaboration stack, right? This space is so multifactorial. It's so multidimensional that they're by necessity, to get the job done for any advanced therapy, requires such a diversity of disciplines and technical capabilities, as well as knowledge, that this will be impossible to consolidate in one place. And most importantly, we've spoken a lot about CMC today, where there's a lot of expertise that is very hard to consolidate in one very small operation. And I think the power of scaling will really lie in strong partnerships and building out the network that is required to deliver the product. But that thinking extends beyond the CMC side of things. Commercializing these products is also a network activity by default, because of what advanced therapies are. It's not sort of pilot, hire, and then have a very nice lean supply chain around this. This by default is a team effort from start to finish, and the finish line at this point is really about how to get these therapies to patients. And as we've seen, that is the biggest hurdle of all of them, is really to scale out from the manufacturing into the market. And that is something that requires, I would say, and that will have to be another discussion of rethinking about who partners with whom to make that happen. We've heard a few examples from prior product launches being discussed here today, but this is a team effort. And so how does innovation look here? It really has to be a collaborative effort in pulling in the expertise that is required to get the ultimate job done, to reach the target, which is to reach the patient, and really be open to having, shall we say, temporal associations, rather than consolidating all this expertise under one roof, because that is unlikely to happen, especially if we're talking about rare diseases.
Brilliant. Thank you so much. I think that really wraps things up very nicely. I am aware it's 3 minutes past the hour. People have lives. You all have day jobs. Lee, I'm sorry we didn't get a chance to go to you to wrap up, but I think what we'll do is we'll aim to send out a wrap up, and maybe even post it online on LinkedIn for everyone to be able to
do that.
see.
That's a great idea.
Let's do that.
Everybody, thank you so much for your time.
Thank you.
Maureen online, thank you very much for your question. We will try and get it answered. I'm sorry we didn't have time to answer it live. But thank you very much, everybody, for joining and for giving up of your time. And I would like to really end off with reiterating our thanks to BCA Advanced Therapies Network, our sponsor for the season, and Vitalant, who's our sponsor for this episode. Without their support, we really couldn't have done it. This session is part of a much larger conversation that we would like all of you to carry forward. Meeting on the Mesas next week. Let's carry on the discussion. Cell and gene therapies hold a lot of promise. Let's make sure that we are in control of them. We develop them. We do the best for patients. Please follow us on LinkedIn. Get involved in the discussion. Contribute to the solutions. And thank you very much, everyone, for joining us. And to my panelists, thank you for your time and for your expertise.
Thank you all. Thank you, Sven.
Thanks for everyone listening.
Thank you,
Bye.
everyone.
Bye-bye.
Bye.